Good day, everyone. On behalf of Cambridge Health Tech Institute's Global Web Symposia Series and our sponsors, Eurofins DiscoverX, a Eurofins discovery company, I'd like to welcome you to biosensor development using CRISPR to quantify endogenous protein modulated by targeted protein degraders. My name is Elizabeth Lamb, and I'm the host and moderator for today's event. Now I'd like to introduce our presenter for today. He is Dr. Chao-Tsung Yang, PhD, Principal Scientist, Research and Development Department with Eurofins DiscoverX. Welcome, Dr. Yang. The presenter ball is yours.
Hi, everyone. Thanks for your time and interest to attend this webinar. In the next 25 minutes or so, I'm going to share you a very exciting new assay platform we developed to look into endogenous protein turnover induced by protein degraders such as PROTAC. Just a very brief introduction of who we are. DiscoverX is a product division of Eurofins. Our headquarters locates in Fremont, California, USA. We offer more than 800 off-the-shelf assays that have been successfully implemented in global pharmaceutical companies, biotech companies, and CRO for their therapeutic discovery and development. At Fremont R&D side, we specialize in cell line engineering, characterization, and assay development. Our assays are based on our core technology, Enzyme Fragment Complementation, or EFC. It is a robust and flexible platform that is well adapted in the industry for functional and MOA-based cell assays. It is a split beta-glycosides system that the enzyme has been broken into two fragments. One acts as an enzyme donor, or ED. The other fragment acts as an enzyme acceptor, or EA. Separated, these two fragments are inactive. When combined, they form an active enzyme. They can hydrolyze its substrate to produce a chemiluminescent signal that can be detected by a simple plate reader instrument. Our assay platform, including G-protein coupled receptor activation and many others illustrating here. Recently, protein degraders have become a new frontier of drug discovery and development. The new emergent therapeutic approach utilizes cellular protein degradation system, namely ubiquitin-proteasome mediated system, to downregulate or deplete overabundant proteins that cause diseases such as oncoproteins in many cancers, or tau protein in Alzheimer's disease. The most well-known example of protein degraders are PROTACs, or proteolysis targeting chimerics. PROTACs contain two active functional groups. One binds to the target protein, the other functional group binds to an endogenous E3 ligase. Upon interactions, PROTAC bring the target protein and the endogenous E3 ligase into close proximity. Subsequently, target protein is ubiquitinated and degraded. The most exciting aspect of PROTAC as a therapeutic agent are the PROTAC could induce protein degradation in a relatively low concentration, and low affinity between PROTAC and the target protein can lead to effective protein degradation. Another class of protein degraders is a class of small molecules that call molecular glues that could bind to E3 ligase and induce a neomorphic activity of E3 ligase to degrade new substrates. One of the best known examples is thalidomide, shown here. Upon its interaction with cellular and E3 ligase, it induces new activity of cellular, and consequently new substrates are degraded, include cell 4, shown here. The realization of thalidomide's mechanism of action opened up a new field to identify new molecular glues that bind to specific E3 ligase and induce overabundant disease protein degradation for therapeutic usage. The current assay to determine the potency and the efficacy of protein degradation, such as Western blot proliferation assays, are tedious, labor-intense, time-consuming, and very low throughput. We believe that ideal assay platform should be compatible with disease cell models expressing physiologically relevant level of target protein and E3 ligase. This platform should be robust, reproducible, and sensitive to detect protein turnovers. It should be also scalable to high throughput screenings. To provide such an assay platform, we decided to use our EFC technology to develop assay platform that could quantify endogenous protein turnover. In our EFC assay, it's a simple and homogeneous platform that is sensitive and easy to implement. To capture the physiologically relevant level of target protein, we knock in a small fragment, EA, roughly about 40 amino acids, into the genomic locus of the target protein using CRISPR technology. We choose ribonucleoprotein or RNP delivery method in which Cas9 protein and synthetic guide RNA are assembled in a test tube and electroporated into the cell alongside with a single-strand DNA donor encoding ED fragment. This is a non-DNA-based method, therefore there is no constitutive expression of Cas9 and guide RNA complex that might lead to off-target effect. This delivery system is easy, high on target editing, and because it is transient, the off-target effect is minimum. In addition, the ED tag doesn't have a lysine residues, which is a residue that ubiquitin covalently links to. Therefore, any target protein degradation we observe would not be the artifact due to the introduction of ED tag. In our assay platform, the endogenous ED fusion target protein produce a modest to high basal EFC signals upon interaction with protein degraders such as PROTAC illustrated here. The ED fusion protein is degraded, resulting in a loss of EFC signals. As a proof of concept, we choose to build BRD4 biosensor in disease cell models for BRD4 and its downstream effector are well studied, and reagents targeting BRD4 are commercially available. BRD4 belongs to a class of protein called bromodomain and extra-terminal motif, or BAT. It is a transcription modulator upstream of c-Myc oncoprotein. Studies have shown treatment of BAT inhibitor leads to decrease of c-Myc expression. PROTACs containing those inhibitors, such as JQ1, also lead to the decrease of c-Myc expression. These inhibitors and PROTACs are commercially available that we can simply obtain to validate our assay. There are several isoforms of BRD4 result from alternative splicing in the C-terminus region. To cover all the isoforms, we decided to knock in our ED tag in the first exon of BRD4. We also generate a c-Myc biosensor with the same approach. As the first attempt, we choose two cancer cell lines that have relatively modest to high level BRD4 and c-Myc expression as our cell models. K562 is a blood cancer cell line, and HCT116 is a colon cancer line. We use a panel of molecules to validate our BRD4 and c-Myc biosensors. They include JQ1, which is a BAT inhibitor, and two PROTAC, MZ1 and DBA1, that both contain JQ1 but engage with two different E3 ligases, shown here, MC1 engage VHLO, and DBAC1 engage with cereblon. We also include a PROTAC control molecule called MC1, which contains JQ1 but doesn't engage with any E3 ligase. This control molecule more or less behaves like JQ1. Our assay workflow is simple and homogeneous. Illustrated here, biosensor cells were first seeded in 96 or 384 cell plates. Degrader molecules were edited and cell with molecules were incubated for desired hours. In our experiments, we try out 5 to 72 hours incubation time. At the end of the incubation time, lysis reagents and detection reagents were added to the same place. After another hour of incubation, EFC signals are ready to be read. We first test out our K562 biosensors, shown here. After 5 hours incubation, we could readily see the on-target activity of two PROTAC, MC1 and DBAC1, with differential potencies. This decrease of BRD4 signal induced by MC1 and DBAC1 is not a result of cell death, as shown here, that no change was observed in the viability assays, consistent with the earlier finding that JQ1 inhibits BRD4 activity but not affect its protein levels. We found no loss of BRD4 signal in JQ1 treatment. For PROTAC control molecules, this MC1, since it doesn't engage with any E3 ligase, it behaves just like JQ1. No loss of BRD4 signal was observed. For C-MYC biosensors, after 5 hours of incubation with MC1, we observed loss of function of EFC signal as we expected. Since JQ1 inhibits BRD4 activities, we could also detect loss of C-MYC expression by JQ1 treatment. Interestingly, we didn't see any downregulation of C-MYC expression by DBAC1 treatment. This is probably due to its low on-target potency at 5 hours incubation. Indeed, after 24 hours incubation, we can detect loss of C-MYC expression induced by DBAC1 treatment. We also noted that after 24 hours of incubation, this molecule displayed some effect on cell viability. As the incubation time extended to 24 hours, 48 hours, and 72 hours, we observed similar on-target potency of two PROTAC, MC1 and DBAC1, suggesting that shorter incubation time is sufficient to determine their on-target activity. However, longer incubation time do increase assay windows. In contrast to our EFC assays, viability assay didn't show much distinct potency between MC1 and DBAC1 until 72 hours of incubation. This table summarizes the MC1's BRD4 on-target potencies and the C-MYC downstream target potencies. The optimal incubation time for BRD4 biosensor is between 5 hours and 24 hours. Longer incubation time doesn't significantly increase its potency. For downstream C-MYC, the optimal incubation time is between 24 hours and 48 hours. Again, the phenotypic assay, endpoint assay such as cell viability assay, longer 72 hours incubation time is required to demonstrate good on-target potency and reach a potential optimal assay window. In conclusion, compared to a phenotypic endpoint assay, EFC-based biosensor is much faster and more sensitive. To further characterize our biosensor, we decided to test additional BAT inhibitor, OTX15, and its derivative PROTAC ARV825. Using c-MYC biosensor, we could generate a rank order of these molecules with differential kinetics shown here in the lab panels. This rank order of potency recapitulates previous published data using c-MYC ELISA shown here in the right panels. Although the rank order is much similar, the potency of those molecules in these two experiments are slightly different. This difference is probably due to different cell type has been used in these two experiments. Nevertheless, the finding of comparable rank order validates the utility of our EFC-based biosensor. A recent publication from Cruz Lab has shown great on-target potency of the NOT-LIN-based PROTAC that target MDM2 E3 ligase. When we test such PROTAC JQ1 endogenous NOT-LIN in our K562 BRD4 biosensor, we find it is less potent than DBAT1 and NZ1. However, when we tested in the HCT116 BRD4 biosensor, we find this NOT-LIN-based PROTAC is more potent than DBAT1. This increase of potency from K562 to HCT116 cell line also hold true in the c-MYC biosensors, which is shown in the bottom panels. The low potency of NOT-LIN-based PROTAC in K562 cells might be due to the low abundance of MDM2 in the K562 cell line shown here in the expression data, while other E3 ligase, VHL, cereblon, show similar expression level between these two cell types, and their related PROTAC also show similar potency between K562 and HCT116 cells. This result suggests that it is important to pick a cell model with relevant expression of the E3 ligase that you are targeting. To demonstrate that the loss of EFC signal in our biosensor is mediated through ubiquitin-proteasome systems, we treated the BRD4 biosensor cells with two inhibitors that inhibit protein degradation pathway at different stages, one at E2 ligase, one at proteasomes. In the presence of both inhibitors, we find greatly reduced potency of MZ1-induced on-target degradation, shown here, suggesting that this assay are truly mediated through ubiquitin-proteasome systems. In summary, we have generated EFC-based assay platform to detect endogenous protein change in disease-relevant cell models. As a proof of concept, we have generated biosensors for BRD4 and his downstream target c-MYC in the blood cancer cell line and the colon cancer cell models. We showed that these biosensors are highly sensitive to detect protein turnover induced by protein degraders such as PROTACs, with more rapid kinetics than a phenotypic endpoint assay such as cell viability. This biosensor line provides a unique opportunity to screen and identify new molecular entities for therapeutic development. Current 4 cell lines we presented here are available through our early access program and will be available through our catalog by spring 2020. Finally, We believe this assay platform should be extensible to other targets and protein degrader molecules. Please contact our custom assay development program. We will develop biosensor for the target of your interest. And also please visit our website for those information. Besides functional assay platform I just talked about here, we also offer various assay platforms listed here for protein degrader discovery and development. Please contact us if you have any interest